Howdy, folks. Thanks for checking into Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this third of our trio on aerobic respiration notes cast, we're going to be talking about the electron transport chain and good old chemiosmosis. Now, to kind of wrap up aerobic cellular respiration here in a nutshell, I give you this diagram. This has got all the important stuff that we really need to remember about the aerobic cellular respiration pathways. Once again, what's the point of this whole process? The point is to make ATP. And now we're going to finally start making a whole lot of ATP. Let's look at how much energy we've made so far. Through glycolysis, we've netted 2 ATP molecules, and through the Krebs cycle, another 2 ATP molecules. And quite honestly, 4 ATP molecules per glucose molecule is just not enough to run us big, huge, clunky animal organisms. Each animal cell is going to recycle over 10 trillion ATP molecules every minute or so, and that's just way more than we're going to be able to do. There's got to be a better way. Of course, there is a better way, and we've been talking about it all along. It's the electron transport chain, that step-by-step sequence of metabolic reactions that's going to take place right along the inner mitochondrial membrane itself. And this is the stage that's actually going to be taking oxygen and giving aerobic respiration its aerobicness. Speaking of mitochondria, let's take a moment to review the mitochondrial structure itself. Remember, it's a big bag shoved inside of a small bag, so we got a smooth outer membrane, and that inner membrane, because it has more surface area, is folded into the cristae. And it's along that inner membrane that we're going to find all of the transport proteins and enzymes for cellular respiration itself. Plus, it's the compartmentalization, that inner membrane space between the inner and outer membrane, that's going to be necessary for cellular respiration. And let's take a little closer look there. It is along these cristae, like I said, that the transport proteins and enzymes are, and what they're doing is actually moving protons from the matrix into the inner membrane space, into that compartment. And it's from this trapped protons that we're actually going to generate the energy needed to make all of those ATP molecules. This is only going to take place if there is oxygen available to act as the final electron acceptor for the electron transport chain itself. And that's what makes this whole process aerobic. Now I'm going to give you some of the details of the steps of electron transport, but it's important for us to remember that we don't need to memorize each step of the electron transport chain. I show you these details because it's important for you to recognize that it's not magic, that it's chemistry, that it's understandable, and that it does actually make sense. So we're going to go into the mitochondria, more specifically into the inner mitochondrial membrane. That makes the space above these proteins here, the inner membrane space, and the space down below that mitochondrial matrix. Now, the first character we're going to meet is complex 1, known as NADH dehydrogenase. The next group of proteins, this is one individual protein, is called complex 3. I know we skipped complex 2. We'll get back to that in a second, but this is cytochrome BC complex. And then the third complex, which is complex 4, is cytochrome C oxidase complex. There's a couple electron shuttles we should be aware of. One is Q here, or coenzyme Q, or ubiquinone. And that's fat soluble. That's going to hang out there in the leggy bits in the hydrophobic part of the membrane itself. And then there's also cytochrome C that is water soluble, but it's trapped in the compartment there of the inner membrane space, and that's going to be another electron shuttle. Throughout the previous 2 videos, we've been talking a lot about the electron carriers from glycolysis making the 2 NADH molecules, and the Krebs cycle making the 8 NADH and 2 FADH molecules. And we always referred to those as electrons or energy in the bank. And well, now is where we're going to actually get at and spend the money in the bank. NADH carries its electron by carrying a hydrogen atom. So when I take that NADH and I break it apart into NAD and hydrogen, remember that hydrogen is element 1. So that means that the hydrogen is really a proton with an electron whizzing around it. So the first thing that's going to happen here at NADH dehydrogenase is that NADH is going to be split into NAD and H. And that hydrogen is then going to have the electron moved through the protein, and the proton that's left behind is going to be shuttled into the inner membrane space. That electron is given to coenzyme Q. Coenzyme Q is then going to shuttle that electron to complex 3, to cytochrome BC complex. And as that moves through that complex, as that electron moves through the proteins of that complex, another group of protons is shuttled from the matrix into the inner membrane space. Cytochrome C is going to move that electron to cytochrome C oxidase. And we're going to move some more protons into that inner membrane space. Now we should talk about FADH2. FADH2 is going to, through complex 2, which isn't even in this diagram, but it's a peripheral protein. It's not integral through the membrane itself, but it's going to transfer the hydrogen from the electron from the hydrogens of FADH2 right to coenzyme Q. The hydrogen is going to be broken down into an ion that is using the electrons moved to pump that proton into the inner membrane space. But because it's skipping NADH dehydrogenase, it doesn't pump as many protons, and so it is a little less efficient. It does contribute, though. Finally, oxygen is going to act as the final final electron acceptor. So a couple of those protons, a couple of those hydrogen ions are going to mix with the electrons, and 1/2 of an oxygen molecule, which is one oxygen atom, to make a molecule of water. The whole process here is building up a proton gradient, more protons on the inside of the inner membrane space than in the matrix itself. What powers that movement of the protons? Well, it's going to be the movement of electrons. As we strip those hydrogen atoms off of the electron carriers, NADH and FADH2, and then the electrons are stripped off of the hydrogens themselves, it's the movement of those electrons that's actually going to power the pumping of the protons themselves. So we get more protons moving into that inner membrane space as the electrons are shuttled from one transport protein, one enzyme, to the other. It's those protons that are going to drive ATP synthase into making more ATP. Oftentimes when we're discussing the movement of electrons, we talk about the electrons moving downhill as they move through the electron transport chain, and that's really, it's not gravity that's making them move. What really it is it's differences in electronegativity. So that electron is less attracted to NADH than it is to oxygen. And each individual electron carrier is a little bit more electronegative. So as that electron is moved to the next carrier, it's held a bit closer and a little bit closer and a little bit closer. And so that controlled oxidation leads to a controlled release of energy. And that little bit of energy can be used to change the shape of the protein and cause the protein to move the proton into the inner membrane space. And there we did it. Now we have finally set up that proton gradient. All right, and it's that proton motive force, now that we have all those protons together, that's going to move those protons through the ATP synthase, through the holes in the membrane, and going to drive the movement of that ATP synthase to actually synthesize ATP through oxidative phosphorylation. We call it oxidative phosphorylation because we're taking inorganic phosphate and we are adding it to the ADP itself. All right, so we are adding electrons to the ADP essentially. So it's oxidizing, making ATP. This whole process of the movement of, in this case, hydrogen ions through a membrane is called chemiosmosis. And that's really the whole point of the electron transport chain is to generate that proton gradient so that it can move back through chemiosmosis. The protons can move through the membrane and move ATP synthase and allow for oxidative phosphorylation. So that's really the connection between electron transport and the production of ATP. After all that detail, here are the four big essential parts of the electron transport chain. First, we're taking those electrons from the Krebs cycle, from the oxidation of the sugars, and moving them into this transport system within the mitochondria. The electrons being moved from proton shuttle to electron carrier to proton shuttle through that whole process, down the electronegativity from one to the next is going to allow for the movement of the protons across the membrane. And oxygen is that final electronegative electron acceptor forming water. As the protons diffuse back through chemiosmosis, back down the concentration gradient into the matrix, they're going to change ATP synthase and allow for oxidative phosphorylation and the production of ATP. Speaking of ATP production and how much, let's do a little accounting here. We get 2 ATP molecules out of glycolysis and about 2 ATP molecules out of the Krebs cycle, and that's going to generate enough NADH and FADH2 to generate, depending on the type of fuel molecules, about 36 ATPs in total. And so we end up with about 40 ATP molecules made per glucose molecule. Some important questions to keep in mind as we are pondering good old glucose plus 6 oxygen yields 6 carbon dioxide, 6 waters, and about 40 ATP molecules are: where did the glucose come from in the first place? And how about that oxygen? Where are we getting that from? And the carbon dioxide? Like, where in this equation do the carbon and the oxygen, does the carbon dioxide actually come from? And where does that carbon dioxide end up? And where are we getting all that water from? And the ATP, can we actually trace where the parts that make ATP come from, especially in this equation that's right in front of us? And this equation is also missing other stuff that is made and raw materials that are needed for aerobic cellular respiration. And then we should really be able to basically connect why we breathe to the cellular respiration process. So what's the point? The point is to make ATP. And finally, we've made all that ATP, and I'm going to end there. In the next videos, we'll be talking about photosynthesis, kind of the reverse of this whole process. But until then, thanks for checking in to Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. If you have any questions, feel free to email me. My address is on my website, Mr. Ulrich's landofbiology.com. Other than that, we'll see you in class.